Welcome to the all viewers and the well-wishers of the world. Purpose of my lecture is learn the knowledge from the word, return the knowledge to the word. Motto of my life is learn, earn and return. I have learned a lot, earned a lot, and now to return a lot to the word. The topic of today is bacterial virulence factors. It is how the bacteria do survive in the host, so we can say bacterial defenses. The ways by which bacteria are capable of survive in harsh environment of the host. Bacteria are hijacked by the protective mechanism of the host cells. Bacteria resist cytotoxic action of the serum, plasma, and the other bloody fluids involving antibody and complement classical pathway or the complement alone alternate pathway or the lysosome, which are all capable of killing the bacteria. So the killing of the extracellular pathogens occurs within phagocytes after opsonization by the antibody and the or complement and phagocytosis occurs, which just destroy the bacteria and escape from the phagocytosis by the extracellular pathogen is a major survival mechanism by which they can survive and infect the host. So the virulence is bacterial weapons against the host. It is also called microbial defenses against the host. It is the quantitative measure of the pathogenicity of the microorganism, whether they are low virulence or having the high virulence, or the quantitative ability of the bacteria to cause disease when introduced into the host in the small numbers. Degree of the virulence is ability of the organism to cause disease despite the host resistance. It is affected by the numerous variables like number of the infecting bacteria, route of the entry into the body, specific and the non-specific host defenses mechanism, virulence factors of the bacterium. So these cause animal death or the illness in a defined period after bacteria are administered by a designated route into the body. LD50, a lethal dose affecting 50% of a population of the animals or the bacteria. ED50, effective dose causing a disease symptom in 50% of a population of animals. Bacterial virulence factors are regulated by environmental factors that control the virulence genes and the common factors like temperature, iron availability, osmolarity, growth phase, pH, specific ions, specific nutrient factors, and bacterial cell density. Genetic and the molecular basis of the virulence. Virulence factor genes are found on chromosome, plasmids, transposon, or bacteriophage. These are the genetic material. These, inte- these integrate or merge into the bacterial chromosomes. Capacity of the Shigella species to invade is encoded on a 140 megadalton plasmid. Heat labile enterotoxin LT1 of E. coli is plasmid encoded. Heat labile toxin LT2 is encoded on the chromosome. Cholera toxin, salmonella enterotoxin, and Yersinia invasion factors are all encoded on the bacterial chromosome. As primary goal of the microbe is to acquire nutrients and multiply, not to harm host, just to live silently and comfortably, not doing to any harm to the host. Bacteriophage infect bacteria and integrates its genome into the bacterial chromosome by a process of lysogeny. Now produce toxins in the pathogenic bacteria. Examples are diphtheria toxin by the Corynebacterium diphtheriae. Otherwise, if these are not going to happen, that diphtheria is toxin is not effective. And the bacteria is not capable of causing the disease. Erythrogenic toxin by the Streptococcus pyogenes, Shiga-like toxin synthesis by E. coli, and botulinum toxin type C and type D by the Clostridium botulinum. Virulence factor has four postulates. Number one, virulence gene or its product must be present. Number two, virulence gene must transform a non-pathogenic bacteria into a pathogen. Just like the Corynebacterium, virulence gene must be expressed during disease process. And fourth is antibodies against gene products are protective in nature. Now, microbial defenses against host. The weapons of the bacteria against host are transmissibility, adherence factors, infecting dose, invasion of the host, invasiveness, communicability, route of infection, serum resistance, toxin or called toxigenicity, and the enzymes. Continuing, the anti-phagocytic factors, phagosome escape, intracellular survival of the bacteria, antigenic or the phase variation, antigenic heterogeneity, antigenic mimicry, iron requirement, plasmids, bacteriophage, and the motility. These are the 18 factors which are considered as the defense against the host, or these are the weapons of the bacteria. Now, why do we write etc many a time whenever we finish any sentence at the end in the exam even? So its mean is that etc, end of the thinking capacity. So I am finished now. Now, the first is transmissibility. It is the ability of the bacteria to be transmitted from reservoir to the host, done by the different routes. It is not present in every bacteria that has the capability of transmissibility. Only pathogenic bacteria can do it. Number two is adherence factors. To avoid physical and immunological removal by the host, the bacteria must adhere to the cell surfaces like in respiratory, gastrointestinal, and genitourinary tracts, solid surfaces on the teeth, heart valves, prosthetic material. Bacteria adhere to the host via pili, capsule, glycocalyx, flagella, liponucleic acid fibers, and biofilm. Other ways are direct interaction, molecular bridging via fibronectin, and binding of the adhesins molecules to the host cell receptors. Here one can see the different things which are capable of causing the adherence, that is LPS, flagella, then capsule, and glycocalyx, curly fibers, and here one can see the pilus. It have got the adhesive tips which just join to the surface of the host cell, and here one can see the host cell, and these tips as one can see are going to attach to the surface and then make the way for entering the bacteria. Similarly, adherent proteins of the bacteria that just attach to the carbohydrates of the tissue cells, and this adhesion cause the infection as the tissue cells are being invaded by these bacteria by the adhesins. So adherence by the bacterial biofilms. The biofilms are formed on the surface by the bacteria as a sticky gum-like web of the polysaccharide to adhere, special bacterial adaptation that facilitates colonization of the bacteria on the artificial valves or the indwelling catheters and the dental plaque. It is protected from the host immune mechanism that it cannot engulf that biofilm. It is also a diffusion barrier to the antimicrobials. So these cannot enter into the bacterial body. These are seen in Staphylococcus epidermidis, Staphylococcus aureus, and Pseudomonas aeruginosa. Here one can see the different receptors of the bacteria and different receptor of the host cell that have got the interaction and that facilitate the adherence, and then it is going to cause the damage to the host cell by getting entry into the host cell. Similarly, one can see the mechanism of adhesion, the different types of the molecules, as one can see the proteins, and that is capable of attaching to the receptor of the host. And similarly, here one can see the biofilm that is being produced by the bacteria, and it have got the sticky nature, and this helps the bacteria to attach to the different prosthetic and valves and other. So that is the biofilm being produced, and that is going to attach there as a web-like or gum-like structure. The third is infecting dose. Number of the bacteria required for the successful infections measured as minimum infecting dose, MID, minimum number of bacteria to produce clinical infection in a susceptible host under standard condition. Then minimum lethal dose, MLD, minimum number of bacteria to produce death in a host under standard conditions, ID50 and LD50. Dose required to infect or kill 50% of the animals tested under standard conditions. Number 4, invasion of the host cells and the tissues. It is also called penetration and spread. Invasion is the ability of an organism to enter a cell or entry into the blood. Invasions are the surface proteins that can invade non-phagocytic human cells, escaping immune system. Virulence factors of the bacteria differ in the different functional types of the bacteria. Some bacterial pathogens reside on the epithelial surfaces like Vibrio cholerae. Some penetrate these barriers, but they infect locally, do not go into the bloodstream and to cause systemic problem. Here one can see the toxins of the staphylococci and then the adhesins. These adhesins are capable of adhering, and then these are the invasive molecules, coagulase, staphylokinase, and leucocidin. I will tell you later. So these are capable of causing adhesion to the host cell and then getting the entry into the host cell via these molecules. Other pass into the bloodstream and from there to the other systemic sites. It is seen in the intestine, urinary tract, and the respiratory tract, less commonly in the skin. Like Shigella penetrates into the intestinal epithelium, cells become endocytic, reside inside the cell, so that is called endocytic, that do not spread into the bloodstream, do not get entry into the bloodstream, but the Salmonella typhi pass through the epithelial cells into the bloodstream. Borrelia burgdorferi is transmitted into the bloodstream through skin by a tick bite. So here one can see the epithelium being invaded by the bacteria and the Salmonella enteritis that get entry into the epithelial cell, and Vibrio cholera just reside on the epithelial cell, but the Salmonella typhi is capable of penetrating the epithelial cell and then it get entry into the bloodstream. So some reside on the surface, some get endocystic living inside the cell, and the some penetrate the cell and cause the spread to the different organs of the body via bloodstream systemically. The fifth is communicability, ability of a microbe to spread from one host to another. No relation exists between virulence and communicability. High degree of communicability may not be exhibited by a highly virulent parasite. So it is the feature of the different bacteria, not depending upon their virulence, it is just their other inherent quality. Infections in which the pathogen is shed in secretion, as in respiratory or the intestinal diseases, are highly communicable. So one can see the communicability, the Shigella bacteria get entry into the M cells, and here we can see it just passes down and then it goes down, and from there it mucosal layer, it goes into the other cells, and here one can see the bacteria is going to the other cells from the basement membrane, getting entry into the epithelial cell, and here one can see they are traversing, moving, and from here they are just going to the neighboring epithelial cell as number 5. So starting from here, coming here to here, number 1, number 2, number 3, here number 4 and number 5. This process is all communicability. Now, 6 is route of infection. Some bacteria cause infection, whatever be mode of entry, such as the streptococci. They are capable of causing infection by any way, any route they can get enter into the body. Certain bacteria are infective only when introduced through optimal route of their liking. Cholera vibrio produce lesion when entered by the oral route, not parenterally. Common routes are mucous membranes, respiratory tract, gastrointestinal tract, genitourinary tract, through the eyes and conjunctiva, through the air. And here one can see 5 major routes of the transmission: contact, direct or indirect, through skin, contact person to person, through an object, that is indirect, droplet, through the respiratory tract, airborne, common vehicle, and vector-borne. Here one can see the different spread by the airborne, fecal-oral, then human feces. It is going to contaminate the food and the other thing. Here again one can see through the fomites, the belonging of the patient, things which the patient do use. Then through the blood, if that is being given, if that is contaminated and not properly cleared, it is also causing the blood-borne infection. So, continuing the route of infection, food is also a route of infection. Water, fomites, objects, skin pores, sweat glands, hair follicles, parenteral, damage to the barrier, puncture, insect bite, cuts, drying of the skin, adherence, attachment to the tissue adhesion, capsules, fimbriae, streptococcus mutans, actinomyces, and Neisseria gonorrhoeae. These cause the attachment to the tissue. Number 7 is serum resistance. To survive in blood, bacteria must resist lysis as a result of the deposition of the complement on bacterial surface, because the complement just deposit on the bacterial surface and just take it to the macrophage for killing. Now, some bacteria destroy complement components that attract phagocytes through C5a peptidase that degrades component C5a, so component cannot be effective to take the bacteria to the phagocyte. Enterobacteriaceae has primary resistance due to the composition of the polysaccharides in the bacterial outer membrane. So, some gram-negative bacteria can lengthen O chain in their lipopolysaccharide to prevent the complement activation and to avoid the phagocytosis. Organisms causing bacteremia like Pseudomonas are less sensitive to killing by fresh human serum containing complement component. So these bacteria can resist the complement in the blood and the serum. So these are called serum resistant bacteria. Now the eighth is toxin, and it is toxigenicity. Toxins help invasiveness, damage cells, inhibit cell function, and trigger immune response. Bacteria cause disease by producing two types of the toxins, exotoxins and endotoxins. It's very easy to understand. Exotoxin is the bomb have been placed and that have been just fired by some device somewhere else, by remote control device. So the bomb exploded by the remote control device is exotoxin. Endotoxin is a suicide bomber who himself having the bomb and causes the damage. So it's very easy. The bomb exploded by the remote control, exotoxin, and the endotoxin is the suicide bomber. Both gram-positive and gram-negative bacteria secrete exotoxin, which are proteins. In contrast, endotoxins are only secreted by the gram-negative, and these are the lipopolysaccharide LPS of the gram-negative bacteria, an integral component of the cell wall of the gram-negative bacteria only, and not in gram-positive bacteria. So, exotoxin can be grouped into functional categories by the symptoms or the disease they produce. Neurotoxin, as the name is, cause damage to the nervous system. Major symptom it produces is paralysis. Clostridium botulinum, progenitor toxin C, tetanospasmin, and Shiga toxin. These are all acting on the nervous system, so called neurotoxin. Enterotoxin, as the name is, these damage to the intestine and tissue of the digestive tract. Major symptom is vomiting and diarrhea. Vibrio cholerae, Chlorogen, Staph aureus, enterotoxins. And here one can see the toxins. The cell wall component is the cell wall component is the endotoxin, and here you can see the exotoxin being produced by the cytoplasm and excreted outside. So these are exotoxin, and you can see the exotoxin structure. It has toxic effect A and binding structure B. Here one can see the two subunits, A subunit and B subunit. The A subunit is basically the toxic substance which is going to cause damage, but the B substance is the binding structure. Here we can see the host cell, the B subunit, do attach to it and get endocytosis, and here it get into the endosome, and in the endosome the bacteria is released. So the A subunit of the bacteria, which is basically the exotoxin, that is released and that causes damage to the host cell. So third variety is cytotoxin. As the name is, these cause damage to the cytoplasm of variety of the cells, damage the cell function, and do cause the cell lysis. The next is membrane damaging toxins. Many bacterial toxins form pores or the holes in the eukaryotic cell membrane. So, hemolysins cause cell lysis, streptolysin O, listeriolysin of the listeria, monocytogenes, alpha toxin of the Staph aureus. These all damage the membrane and making the hole in that. Here one can see the exotoxin. These are just going to attach to the receptor, and here again you can see the receptor for the exotoxin. They do attach to the receptor and do not come in contact with the antibodies, and just the receptors are going to make hole and the pores, and through which one can see the host cell is being invaded by the exotoxin. These are the exotoxin, as one can see, released by the bacteria, get entered into the host cell and causes the death of the host cell. Now, endotoxins are LPS of the gram-negative cell wall, not present inside the cell like the exotoxin seen by the gram-positive bacteria. These are produced by gram-negative bacteria only. These are labile, so not suitable for use as toxide. Lipid irresponsible for the toxic properties, that is a part of the LPS. Symptoms are due to the vigorous immune response to the LPS. Toxin responsible for septic shock, also known as endotoxic shock. Most of the effect of the endotoxins are mediated by the tumor necrosis factor, and the treatment is using the anti-endotoxin or anti-TNF antibodies to just kill this TNF and neutralize that. Here one can see the endotoxin responsible for releasing the histamine, tumor necrosis factor, activation of the coagulation system, release prostaglandin, thromboxane, leukotrienes, prostacyclin, release beta endorphins, bradykinin, free radical, platelet activating factor, C5a, C3a, myocardial depression factor, leukotriene A and interleukin 2. These are all responsible due to the effect of the endotoxin on the different cells and parts of the body.